Hello, anatomy students. Welcome back to Unit 5, Module 2 lecture video. This is about cerebrospinal fluid. These are your objectives. After this video, you should be able to describe how cerebrospinal fluid, or CSF, is made, explain the functions of CSF, and tell me how it circulates through and around the brain and spinal cord. Tell me how CSF is returned to the venous blood supply, and describe how and why lumbar punctures are executed. So we have spaces that are filled with fluid, cerebrospinal fluid, that are found in the brain. We have two lateral ventricles, one in each cerebral hemisphere. We have the third ventricle, which is in between them, which actually surrounds the thalamus. And we also have the fourth ventricle, which is found between the brainstem and the cerebellum. The third and the fourth ventricle are connected to each other by the cerebral aqueduct. Note that in this picture, we're looking at a brain as if we were looking directly into someone's face. So we're looking at the anterior view of this brain. The formation of cerebrospinal fluid takes place in the choroid plexus. The choroid plexus is a collection of capillaries that is found inside of the ventricles themselves. These capillaries are projecting from the pia mater into the ventricles, because remember that the pia mater are going to support small capillaries. The blood that runs through these capillaries are pushed out of the capillaries and through the ependymal cells, which, if you recall, are a type of neuroglia. And then the filtrate goes into the lateral ventricles. Now, coming from the blood, you might think that the filtrate is the same as blood plasma, but it does have a couple of differences. For instance, blood plasma is very high in proteins, while the CSF is almost protein-free. And then there are some ion differences between the two as well. The function of cerebrospinal fluid is not only to cushion the brain in order to keep it from mashing against the inside of the skull, during trauma or during a concussion, but it's also going to help to inform the brain of the body's internal environment. So pH is a measure of hydrogen ion concentration. And carbon dioxide dissolved in the blood is going to directly affect the amount of hydrogen ions that we have in our blood. So the more carbon dioxide that's in our blood, the more acidic our blood is. So if you up the carbon dioxide, that means that you're increasing the amount of hydrogen ions, and it means that you're decreasing the pH, making it more acidic. Well, all of these ions are going to filter into the cerebrospinal fluid as well. And then as the cerebrospinal fluid circulates throughout the brain and the spinal cord and around the outsides of these, there are different areas of the brain, nuclei, that are going to sense changes in pH, typically caused by carbon dioxide levels. So that can trigger some homeostatic processes in the brainstem. Recall that the brainstem, the medulla oblongata specifically, is responsible for respiratory rate. So if you have an increased amount of carbon dioxide in your blood, maybe because you're exercising at the time, that means that your brainstem is going to sense this change and cause your respiratory rate to increase so that you are exhaling more and more and more carbon dioxide. So that's just one example on how the cerebrospinal fluid helps to inform the brain of what's going on in the internal body environment. So this picture looks a little bit scary. Please just label the numbers on your paper as I talk through it. So what we're looking at is a coronal section of the brain and the spinal cord. And so this darker portion here is the cerebral cortex, and the tan portion is the white matter. And then we have our brainstem, and we have the spinal cord here. And remember that the meninges are going to extend beyond the spinal cord. So that's what this dip in the meninges is here. So number one is pointing to the lateral ventricles, one in each cerebral hemisphere, and they're connected to each other by the interventricular foramen. That is going to form into the third ventricle, which surrounds the thalamus. So third thalamus, that's the way I remember it. Number four is a tiny canal called the cerebral aqueduct, and that's going to connect my third ventricle with number five, which is my fourth ventricle in between the cerebrum and the brainstem. And then you'll notice number six. We have little holes here, foramens, which if you should try and remember from the skeletal system unit means a hole. So they are lateral to the spinal cord. So there's one on each side, but your book calls them the lateral apertures. Either way, they're just holes and the cerebrospinal fluid is flowing through them. Number seven is pointing to the medial foramen or the medial aperture. And you have to think of number seven as allowing the cerebral spinal fluid to flow like out at you. Like imagine this as being three-dimensional. And so the cerebral spinal fluid is going to bathe the outside of the spinal cord by coming out of the fourth ventricle from the medial foramen slash aperture. So number eight is the subarachnoid space. And remember from the last video that the cerebral spinal fluid is going to circulate below the arachnoid mater. And if you look up at the top here, number nine is pointing to a structure called the superior sagittal sinus. In the last video, I mentioned that the in between the dura, the periosteal dura and the meningeal dura have what's called a potential space because you're not supposed to see it unless there's been trauma or death. However, there are three areas in the brain where there is separation of these two membranes of the dura mater, and they form what's called the dural venous sinuses. One of the dural venous sinuses is the superior sagittal sinus, and that's what we're looking at up here at number nine. And so this is one of the dural venous sinuses. And then there are two others. But what these spaces do is they collect or like pool blood from the veins, and it's in this blood that circulated cerebral spinal fluid is going to enter and then get dumped into the venous blood supply and get circulated throughout the body again. So the dural venous sinus is just a space in between the two layers that form the dura mater, and it's filled with venous blood. So now we're looking at a different view of the brain. We're looking at a mid-sagittal section. So this is the inside of the brain itself. No, go ahead and label this picture. I put this on the handout for you. Number one, we're looking at the lateral ventricles. And so like this fluffy pink looking like part, this is the choroid plexus. And you can see that we have tiny blue and red blood vessels in there that those are the blood vessels that are flowing through. Number two is showing us the interventricular foramen. And so recall that this is a hole that's going to connect the two ventricles, the one on the left with the one on the right. Number three is the third ventricle. Number four is the cerebral aqueduct. And I think that here you get a better feel for how the cerebral aqueduct is connecting the third ventricle with the fourth ventricle, which is labeled here at number five. And now remember that like this whole section is actually a solid, well, not solid because you have the canal running through, but it's a tube, it's a wall. And so number 6 is showing us the hole, the lateral foramen slash aperture that's on the other side. So the cerebral spinal fluid will flow down here and then it'll exit out this hole and then circulate in the subarachnoid space around the brain and the spinal cord. So that's number 6. Number 7 is showing us a really good picture of what the medial foramen would look like. And so here you could have some cerebral spinal fluid would enter out the lateral aperture. Some of it will continue on down the canal to number 7 to the medial aperture, and it'll exit here, and it could go down and on the outside of the spinal cord, and that's what this little arrow is showing us here. Or it could go around the cerebellum and around these other structures of the brain. So all of this pink area in this picture, this is our subarachnoid space. That's what the number 10 is pointing to. Oh, but I digress. Number 8 is a little portion that is called the cisterna magna. This is at the very base of the cerebellum, just outside of the medial foramen slash aperture. And this is just another space. And of course, just like everything in anatomy, everything has a name. This is called the cisterna magna. Number 10 I mentioned. And then number 11 is here. And this is an interesting little structure. I think that they look like little chef hats. But these are called the arachnoid villi or the arachnoid granulations. Your book is calling them the arachnoid granulations. So I wanted to include that on the slide here. This is a blow-up picture of the arachnoid villus slash the arachnoid granulations. And you can see that they kind of are tiny little extensions from the subarachnoid space. And notice that they are kind of penetrating into or extending into the superior sagittal sinus, which remember is just a separation of the periosteal dura mater and the meningeal dura mater, and it's filled with venous blood. And so what happens is that cerebral spinal fluid will flow through here. It'll go up into our arachnoid villus slash granulation, and then it will diffuse into the venous blood that's filling our superior sagittal sinus. And from there, it's going to enter venous blood supply. And so it's just getting recycled. All right, so this is a picture of a lumbar puncture. And remember again that the meninges extend beyond the spinal cord itself. And so in the lumbar portion of our spine, so here's the third lumbar vertebra and then the fourth one. And then this is the space that's in between the two vertebrae. And what they're doing is they're inserting a needle here. Now, the reason why is because this is the end of the spinal cord, and then it kind of starts to branch off into nerve-like extensions. And so if, and I'm just going to hold my fingers up here, so if a needle were to go through, it would just cause, like, push aside those fibers without damaging them at all. And so that's why this works. So a lumbar puncture is usually used to collect cerebrospinal fluid in order to test for abnormal characteristics, like to see if it's cloudy, to determine the pressure, the amount of cerebrospinal fluid that is inside. Or if they need to see something inside of the spine, they can inject some dyes for imaging. All right, we don't have a lot of time for questions, so